We're in the hop yard at Borderview Research Farm in Alburgh, Vermont, and today is August 28th, 2020, and we're about to start harvesting hops. John, what type of hops are these?
Yeah, so today we're just harvesting our Centennial hops. We started 2 days ago, and so in this process we're just going to kind of go through, clip the bottoms of the bines, and then afterwards we'll go through on the lift and cut the tops and kind of lay them out by plot. So we're just trying to collect some data based on our fertility study that we've conducted for the past 2 years here.
Great, so let's take a look at the equipment.
So we're just standing in front of the lift that we use to harvest hops. This is where I'll hop up in the basket and Travis will give me a lift. So we'll get up to the top, cut the tops off, and lay them across the bars here by plot. Travis has been doing this for 5 years or so.
5-6 years, something like that.
Yep. And so we also use this to tie the quar up to the top wire in the spring. So once we've cut all the bines down, we bring them back to the barn and we start to lay out all the bines to run through the harvester. We'll kind of load this up by plot and run one plot at a time for our research. And then we'll start feeding things through our mobile hop harvester, which Travis generally operates as well. So I'll help him out a little bit, and I'll just kind of grab a vine one at a time and then place them on the hooks to run through.
So I'm Sarah Ziegler with the Northwest Crops and Soils team, and today I'm out in our no-till cover crop experiment where we're looking at spring manure application versus fall manure application under conventional tillage and no-till systems with and without cover crops. And today I'm out here to take populations because this corn is getting close to harvest, so we want to know how many plants per acre are actually out here versus what we planted at. So if we need to make an adjustment in our yields to have a standard population, we can do that. And also I'm taking corn stalk nitrate tests. And so this test, I'll show you how to take one, but we're really using that sort of like you would use a pre-sidrus nitrate test to see how much more nitrogen you needed for the corn to mature. This is sort of after the fact, how much nitrogen did the corn actually take up. So it's an indication for our research of how much nitrogen was actually made available given the different cropping systems that we're looking at. And so one of the things we're interested with this project in particular is understanding how to integrate manure into a corn silage system. So I'm taking the corn stalk nitrate test today because we're interested in looking at the difference between these different cropping systems and how much nitrogen is actually available to the crop. So in a no-till system where you're not actually incorporating the cover crop if it was there, or the manure when you spread it, you have a lot less nitrogen becoming available to your crop because you're applying it on the surface, you're not introducing all that oxygen and microbial activity to breaking it down. So if you're limited in your nitrogen availability, is your crop going to suffer if we're not adding additional nitrogen here, so we can see that effect. So we can see what sort of yield decline we may see in that kind of a system, and it's trying to really get a better handle on how we integrate manure into a system given those different tillage options when you also have a cover crop kind of thrown into the mix, and what those trade-offs are in terms of yield, some of the barriers to being able to do that, and those kinds of things. So that's what I'm doing, and I will show you how to take one. Okay, so to take the corn stalk nitrate test, what I'm going to do is, this is our plot here. It's 4 rows of corn and it's 40 feet long. So we're going to take probably 5 different corn stalks throughout this randomly. And what you want to do for the nitrate test is it's taken from the bottom of the plant. It's an 8-inch segment, 6 inches off of the ground. So you're essentially taking, when you would go and chop your corn, you're essentially taking that bottom 8 inches of what would get chopped. And that's what's analyzed for nitrate nitrogen. and there's basically a scale of the nitrogen content that we'll see there, whether or not that's sort of a low concentration, indicating that there wasn't sufficient nitrogen to that crop, all the way up to something that would be excessive, kind of like what you'd see on a soil test. So what I'm going to do is go in here and just kind of randomly stop at a plant. And then I've made my handy little measurer where I'm going to go to 6 inches off the ground, and I'm going to cut it. And then I'm going to try not to get soil on the tip of it, because that'll can influence the nitrogen content with the analysis. And then I'm going to measure the 8 inches, and I'm going to cut this segment. And so I'm going to do that through this whole plot 5 times. Usually these have to be dried and ground for analysis. You can also usually send them into a lab wet, but we usually dry them, so to make it a little bit easier to dry that corn stalk, we usually also end up quartering them or cutting them in half. But I'll just throw it in the bag, keep on going, and when we get 5, we'll be done in this plot.
Hi, I'm Lindsay. I work for UVM Extension Services as a research specialist, and today we're out here planting the Precision Sustainable Agriculture Common Experiment 1 trial. And in a nutshell, what this is looking at is 3 different kinds of cover crops: winter rye, a hairy vetch, and a mix of those two versus a control to see how they affect soil dynamics. In particular, nitrogen. We're also looking at phosphorus. We're going pretty far down in the soil profile, about 3 feet, to see what those changes might be. After we terminate the cover crop, we'll be planting corn silage in this area, and in between the cover crop treatments, we're going to have nitrogen sub-treatment, so 6 different rates of nitrogen to see how the cover crop and the nitrogen affect the crop quality and yield. So one of the things that we'll be looking at as the corn crop grows is cover crop decomposition and how that affects nitrogen availability in the soil, and then also therefore the crop response to that. And so these litter bags are going to be filled with a quantity of the cover crop and then just the left out in the plots. And we'll be collecting those at different intervals, 1 week, 2 weeks, 3 weeks, so on, and then analyzing that cover crop to see how much nitrogen has left it over time. And so with this, we'll be able to see at what point in time is the biggest release of nitrogen from a rye crop, from a vetch crop, and from a mix. And so we'll be able to see if there's a steady rate or if that a flux of nitrogen happens right when our cash crop, corn silage in this case, needs it most. Part of this trial is that it is in collaboration with a number of other states. So we are one of 15 states going all the way from Florida to Kentucky to Vermont and further out west. And we're really excited to see how the variability in nitrogen and cover crops changes from region to region, so we can make better recommendations for our farmers.